Hello everyone.
Hello.
Hello. How are you doing guys today?
Mhm.
Good?
Good.
Good.
Good, perfect. Can I ask you one more thing before we start? I would like to uh, kindly greet you in Japanese, but can you maybe correct me if I'm pronouncing it incorrectly? Is it konnichiwa?
Yes.
Konnichiwa? Okay,
Ye-
thank you very much. So,
yes.
konnichiwa to everyone. And okay, guys, so today we are going to start with or we are going to continue, better to say, with cell organelles, cell membrane, and if we have enough time, we will see how we will handle the time management. We will continue with cell membrane transport. Um, can I ask you also, before we started, have you checked the Google Classroom post yesterday evening, my time? I posted a few videos you could check or have you seen that? Some of you, I'm not checking whether you've seen the videos, only the post, whether you saw that I posted something. So you are aware that there is something in the Google Classroom, right? Can you just thumb up if you saw the post?
Yes,
Yes,
I checked.
perfect. Okay. Okay, ladies and gentlemen, there are few videos in the classroom I recommended to watch. If you didn't have enough time, it's absolutely okay. We will go through that together. Nevertheless, yes, there is a fluid mosaic model, biology, cell structure, uh, inside the cell membrane, and cell transport. We will go through all of these topics together. Um, just maybe what I will recommend, maybe after this session or tomorrow's session, it will be good if you watch those short videos. I will tell you direct, especially which of them will be not necessary, but very beneficial for you for a recapitulation of what we discussed today. Okay, so are thank you very much that you are following the posts I'm posting to the Google Classroom. And yes, I think we can start. Do you have any questions before we start? Mm,
No.
no?
No.
Everything's okay, so can we start? Okay, perfect.
Okay.
Okay, guys, so let's start with a little introduction. Some warm-up activities prepared for you, so I suggest that we can start. Yuki, please, can you read the question and also the options, please?
Um, which of the following statement is true about prokaryotic cells?
And also the options, if it's possible.
Uh, they, they lack a nucleus. They have membrane-bound organelles. They are larger in size compared to eukaryotic cells. They are only found in plants.
Thank you very much, Yuki. Very good. So, guys, have maybe 15 more seconds to read it one more time, and then we will do a poll. Okay, let's do a poll. You can also um, place the poll table all around the display so you are also able to read. Okay. Okay, so here are the results. Let's discuss them. Hmm. Okay, so Yuki, can I ask you, what have you chosen and why? Can you explain why? You can try.
Yes. Mm,
What option have you chosen? A, B, C, or D?
A.
A. Hmm. Okay, and can you explain why you think A is correct?
So I think prokaryotic cells have DNA, but so they don't have a new nuclear.
Hmm. Hmm. Nucleus or nucleoid.
Yes.
Hmm. Okay, very good, Yuki. Thank you very much. Um, okay, so Yuki went for A. Is there anyone who went for B, C, or D? Hmm.
I
Misato,
went
can I
B.
ask you? Um, do you hear me, Misato?
Yes,
Okay. Uh, can you explain what have you chosen? First question. What have
I I choose B.
B? Okay. Can you
Yes,
explain why B you think is correct?
yes. I cannot realize membrane bond organize. I think membrane bonds is
Hmm.
double
Double
bond.
layer?
Yes, I
Hmm.
think double
Hmm.
layer means yes.
Membrane-bound organelles. Let's show me the picture. Okay. So here we are back to the prokaryotic and eukaryotic cell. Membrane bound organelles means it will be better on this picture when we are talking about eukaryotic cells. Um, Membrane-bound organelles such as endoplasmic reticulum, we are able to see here, or nucleus, or Golgi apparatus. Those organelles have their own membrane on the surface, on the top of them. And so this is these membranes are separating those organelles from the cytoplasm or from the surrounding inside of the cell. So when we are talking about membrane-bound organelles, we are talking about mentioned organelles, not about plasma cell membrane. We were talking yesterday, which is the phospholipid bilayer. Phospholipid bilayer is a cell membrane. Okay, cell membrane.
Okay,
Let me get back here. So we are able to see plasma membrane or cell membrane. Both terms are correct. Yes. And this is a plasma membrane, and this is a phospholipid bilayer. Yes, and it's on the surface of a cell, and plasma membrane separate or also protect the cell itself from the surrounding environment. Okay, so when we are talking about membrane bound organelles, we are talking about organelles in eukaryotic cell, inside of the cell, inside of the cell, and plasma membrane is on the surface, on the surface of the cell, and it separates it from the environment around the cell. Okay, so
okay.
we discussed why B is not correct, right? Now, clear, Misato?
Yes,
Okay, so prokaryotic cell doesn't have membrane-bound organelles. Okay, I think it needs a little bit more clarification because here we do not have such organelles like endoplasmic reticulum, Golgi apparatus. Okay, as you see, there are some ribosomes, plasmids we discussed yesterday, and also DNA. However, they are just swimming, they are just flowing inside the cytoplasm. There are no membranes and also no cytoskeleton inside. So it's more loose, let's say, in the inside of it. Okay?
Okay.
Okay, perfect. So we discussed and we clarified why B is not correct. Very good. What about C? Now, what do you think?
Ah, me?
Yeah, yeah, now.
Ah, C is not correct, I think, because prokaryotic cells is smaller than eukaryotic cells, so this is not correct.
Very good, exactly. Yesterday we saw a picture when we were comparing prokaryotic cell. Yeah, this is a Escherichia coli, this is a bacteria yesterday we saw, and here we see the size difference between prokaryotic cell and some examples of eukaryotic cells, like red blood cells or lymphocytes. So definitely here we are able to see, here is very obvious and apparent that prokaryotic cells are much, much smaller comparing to eukaryotic cell. Mhm. So this is the also not correct statement. They are larger in size. Mhm. Okay. And what about D, Sora? What do you think?
Okay, I think D is not correct
Okay.
because prokaryotic cells only have bacteria.
Uh, do you mean that only bacteria belongs to prokaryotic cell?
Yeah.
Sorry, that's my phone. I will just switch off the sounds. Um, One more thing belongs to prokaryotic cell. Yesterday we were talking about that. You are right. You are right. They are only found in plants. That there is one more thing, archaea, what belongs to prokaryotic cell. Yesterday we were talking about few differences which are possible, if you remember. So I will just remind you very quickly that archaea is a different group, separate individual group of prokaryotic cells. They were you would be right, Sora. You would be right before 1977. They were just one group of prokaryotic cells. They were only bacteria. Nevertheless, after 1977, the scientists decided that they will be two separated groups, and there are some differences. Archaea are uh, more adaptive, or it's much more likely that you will find in extreme environments archaeas cells. Also bacteria, you are able to find them, but archaeas are more adaptive for extreme environments such as hot springs, high temperature, low temperature, very acidic environment. Yesterday we were talking about this. So yes, you were right that D is not correct. You were right about that. Just one more thing I needed to mention additionally, to bacteria you mentioned. Okay, so also archaeas.
Okay.
Mhm. Okay,
Uh,
so we are able now to see that which of the following statements is true about prokaryotic cells. Um, they lack nucleus. Guys, maybe one more thing regarding the option. Um, what was also meant by this option was this, that bacteria or also a prokaryotic cell. One second, there is an echo. Okay, there is no echo anymore. Bacteria and also viruses and also archaea. So most of these very small living organisms live also inside of the multicellular organisms or larger, bigger, bigger organisms like humans or plants, fungi, uh, other animals. For example, there is what is meant by that? In your intestine, there are more bacterial cells than the number of your human cells in your whole body. So in your gut, in your intestine, there are more bacterial cells than all of your cells, eukaryotic cells or human cells in your body. So, and they live inside the other organisms. This is also why D is not correct. They are only found in plants because prokaryotic cells can be found also in other organisms such as humans, fungi, and so on and so on and so on. So, do you understand now? Clear?
Yes.
Perfect. 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 Um, very good. Okay. Yuta, please, can you read? Yuta, do you hear me? Uh,
Okay.
yes, perfect.
One liquid layer is typical for animal cells, plant cells, ar- archaea, archaea,
Yeah.
eukarya,
Okay, so eukarya is D, C is archaea.
eukarya.
So please you have 10 more seconds to reread the question and then we will do vote. Paul, sorry, Paul. Few more people need to answer and then we will start. Okay, perfect. Okay, here are the results. So let's discuss them. Okay, 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 okay. So, Yuta, what have you chosen and why?
I chose D.
The eukarya. And why?
Mm, I think uh, eukarya. Uh, sorry, I can. I don't have a reason.
You are not sure why D. Okay. Um. Okay. What about the others? Aoi. Aoi. What do you think? Or what have you chosen? Do you hear me, Aoi?
Yes, but I guess you can't hear me, so.
Yes. Now we can hear you. Now we can
Oh,
hear you.
really?
Yes. Yes. Now yes. You.
Oh,
Okay.
last time you
You
couldn't hear, so I thought,
yeah.
yeah.
You have to just unmute yourself. Here up there
No,
on
I
left.
think I am muted.
Yes.
Yeah, it doesn't matter.
Okay,
Uh,
so what have you chosen and why?
uh, I chose D.
D, and why?
Mm, I thought it's not for only animal or plant cells, so
Hmm.
I chose D.
Okay,
Yeah.
okay. So there are two Ds so far. Shoko, you are raising hand. You have to unmute yourself first. Mhm. There is an
Ah,
yes.
okay.
Now
All
you are
right.
unmuted. Yes, we
Oh,
can hear you. We can hear you now.
okay.
So, what have you chosen and why, Shoko?
I choose A
A.
because we know animals cell members is uh, monolipid by layer,
Hmm.
but I'm not sure about that. About eukaryote has membranes, but is it monolipid by a layer? Is it equal?
Um, Sorry, there is a typo in the question. I just noticed now. It can't be monolipid, but phospholipid. That's my bad. I will immediately try to. No, 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 no. That's typo, and that's my mistake. I will wrote it. Phospholipid by layer. Okay. This is a correct, not monolipid, phospholipid bilayer. Okay, phospholipid means I one more time apologize for that. Phospholipid means the chemistry or that there is some phosphate group and also a lipid part of the molecule. And we yesterday talked about bilayer. Basically, there are two layers.
Mm-hmm.
Uh-huh. Okay, so you went for A.
Mm-hmm. Yes.
Okay.
Like,
Why A?
because animals everyone know animals has
Mhm. Cell
two,
membrane?
yes, cell membrane, and it that cell membrane is two has two layer.
Mhm.
So sorry, this question is mono.
Yes, but this is sorry. Don't check that. This is my typo here. Okay, we are asking about phospholipid bilayer.
So
Phospholipids. So this is
this is a one layer.
mhm.
So I mistake.
You are not mistaken.
No.
And the
Mm-hmm.
the this was question where are more answers correct, and I intended to put it here whether you are able to notice there are more answers correct, because also in your admissions exams there will be some questions which have uh, more options correct, and you have to notice them even though it's not stated before in advance. Some questions have only one option correct, some questions have more options correct. So,
Mm-hmm.
uh, what's meant by that basically is that in this case you were right, guys. Eukarya, uh, but also animal cells is correct answer in this case, and also plant cells is correct in this case. And archaea is very questionable answer, because even archaea can be right in some cases, but for archaea, it's not rare that some archaeas have a, phospholipid monolayer. Yesterday we were talking about that. If you remember that we were talking that every cell has a cell membrane. Yesterday we were talking about that. And what we were talking about, Shoko, is that we have this structure in cell membrane, right? Phospholipid bilayer,
Yes.
basically. There are two layers of phospholipids. And this is something what almost all of the cells have in this structure. All of the cells have cell membrane and most of them have phospholipid bilayer. But some archaeas have just one layer of it, and that's
Um,
one of the factors why are they able to survive and also reproduce and colonize in such extreme environments. Okay, so in this question were at least three answers correct. Okay, guys, so please be aware of that. I know that you, not all of you are going to attend just one specific university. I think in Masaryk Foundation or Masaryk University in Brno, there is always only one question correct, but I have to check that. I'm not sure. I'm from Charles Foundation, and I know that in Charles Foundation there are also multiple choice questions, and that it's not stated. So you have to always read all of the answers, and if there are more answers correct, you have to cross all of them. Then you, if not, you will, if not, you will receive. Not full points. So just think you need to be aware. Still, you have 1 year for preparation for admission, right? Am I right now, or not? You are going to prepare for admission entrance exams, right? Or not?
Yes.
Yes, yes.
Uh,
Yes, so you will discuss that. You will discuss that. You will be prepared. You will be trained for that. Just yes, this is something. This question is something you will have in this way. You will face. So please always read all of the options and be sure that you are crossing all of the correct options when you are dealing with question. Okay, so this one was tricky, but yes, admission questions also be tricky. But very good job, guys, so far. Perfect. So 3 of 4 answers were correct in this case. Okay. And why C isn't is that archaea can also have mm, phospholipid monolayer. That's why phospholipid bilayer is not typical for archaea. Also, monolayer is typical for archaea. So that's why we didn't went for C. Okay? Perfect. Thank you very much. Yuiko, please, can you read?
what is a plasmid? A. A small, circular piece of DNA found in bacteria. B. A large, linear strand of DNA found in animal cells. C. A protein responsible for cell division in plants. D. A type of virus that infects bacteria.
Thank you very much, Yuiko. So again, guys, let's have 10 or 15 seconds and for rereading, and let's do a poll. Okay, one more person. Okay. Okay. So here are the results. Let's discuss them. Yuiko, what have you chosen and why?
Uh, oh, I chose B, but I don't know why I chose.
Mm-hmm. So it was more a guess?
I think I thought A or B, so I choose B
You choose B.
finally.
Mm-hmm. Okay. Okay, Satsuki, what about you?
I chose A
A.
because
Mm-hmm.
plasmid is a fragment of DNA, so C and D is not correct. And plasmid is particular to bacteria, so I chose A.
Mm-hmm. Very good. And you also explained the other options very well. So exactly, yesterday, just remind me what we were talking about plasmids. Okay, so plasmid, here we are able to see, you mentioned also the fragment of DNA. I don't think it's a absolutely correct definition, but I appreciate you mentioned the the thing I told you. Nevertheless, you are right. Plasmids are small circular DNAs which are which are located and are just swimming around inside the cytoplasm. We were talking about their function, that they are not essential for cell itself for surviving, but they can be very beneficial and helpful. And we mentioned the example of antimicrobial resistance, for example, that this antimicrobial resistant information can be stored in those plasmids, for example. Here is very nice picture, small circular DNA. If you remember it as a fragment, I don't think it's something wrong. However, on the admission questions will be stated something like this definition: a small circular piece of DNA found in bacteria. Also very important, except small, is to mention circular, because yesterday we were talking about prokaryotic cell DNA, that and we already today mentioned that there is no membrane or capsule around the DNA, so it has no nucleus, and we have only nucleoid or circular DNA. So this is the same what applies for plasmid. It's just small fracture or fragment of circular DNA located in the cytoplasm of prokaryotic cells. So very good, Satsuki. Thank you very much. So that was that. It's definitely not type of virus, as you mentioned. Sorry if I use red here. I don't know. Sorry, my bad. So small and circular piece of DNA found in bacteria. It's also not a protein responsible for cell division. And what's wrong with B? Mona, do you have any idea? Mona, do you hear me?
Yes,
Okay, perfect. Can you maybe try to explain why B is not correct here on in this case?
because
Mm-hmm.
plasmid is very small
Mm-hmm.
and not only animal cells, I think.
Yes, it's not in animal
From
cells.
bacteria,
It's much more typical for bacteria here and prokaryotic cells. So exactly, it's large. Also, this statement. Okay, again. sorry. It's not large, definitely. It's not also typical very much for animal cells, and also linear strand of DNA. Okay, it's a circular. Yesterday we were talking about that and we just saw a picture. Very good. Okay, so this is a circular DNA and it's nucleoid. Mhm. Okay, okay. So guys, that was it. That was the little introduction. Thank you very much for participation and also active for your activity. Uh, and that was great. And now that brings us to the core of the session. Um, Before we start with the core, guys, we have 14 minutes before first break. So again, I would like to vote if you want to have a break today or not. Okay, so again, yes, I want a break. No, I don't want a break. Okay, one more person. Phew. Okay, so I don't know what to do now, guys, because it's a draw. Uh, okay, so let's have uh, let's let's start. Okay, let's start with our program and with our agenda. And at 8:49 at 8:50, I will ask you again. You will see how much energy you have. If you will handle it without break, or you will need a break. Okay, so in 30 minutes, I will ask you again. Okay, guys, so let's talk something about eukaryotic cell, and then we will get to your homework activity you were supposed to prepare together. So let me have few statements regarding eukaryotic cell. Let me just Okay, all of you are able to see the slides, right? Do you see the slide? Perfect. Okay, so I have to turn it on here. Okay, so regarding the structure of eukaryotic cell, we were already discussing that eukaryotic cells are larger in the size in comparing to the size of prokaryotic cell. We saw it today already. Here is one of the example. Nevertheless, red blood cells and lymphocytes are just a few examples of eukaryotic cells. Some eukaryotic cells can have even could be even bigger. So usual and very typical range for eukaryotic cell in size is something between 5 to 100 micrometers. Okay, I will draw it here. 5 to 10. Okay, 5 is pretty small, but 100 micrometers is very usual for eukaryotic cell. Okay, so we already discussed that why it is called eukaryotic, because it has a true nucleus. When you are going to deal with the Latin during your first year in medicine, you will understand why. Because you means something what is true, or it's your own. We will not bother us now with the Latin. Honestly, or my personal opinion, I didn't like Latin at all. Nevertheless, it's something we have to do. You have to deal with, especially at universities. For some people, it can be very interesting, and yes, also I found something very interesting, but plenty of things you will deal with. You will decide whether you like it or not. So yes, let's move further. Um, we discuss also the nuclear envelope, that nucleus is then separated from a cytoplasm. Mhm. Chromosomes. Chromosomes are always multiple and have a much more complex structure than the prokaryotic chromosome or the DNA. eukaryotic cells are comparing to prokaryotic cells, very rich in membrane system, and we will get there very shortly. Um, this membrane or endomembrane system divide the cell into separated compartments, let's say, or divisions, we can talk. Um, Basically, thankfully, those membranes inside of the cell, uh, these membranes separate those organelles into their compartments or divisions. So here we are able to see a compartment of endoplasmic reticulum with a nucleus. So nucleus continuously is going or connecting with endoplasmic reticulum, and then different organelles such as Golgi apparatus have its own membrane, and we can now divide the cell into Golgi apparatus compartment or endoplasmic reticulum with nucleus compartment or division. It's not important the word we use. What is important to know how to understand or imaginate or comprehend the meaning behind. What is very good for these organelles is the video I uploaded for you yesterday about the cell. It's, I think, 7 minutes long. If we will have enough time today, we can watch it together. However, if not, I think as a recap of today's session, it would be great. You have it. You have it in the Google Classroom. It's a biology. The video name is Biology Cell Structure, and it's 7 minutes long. Okay, so after the session, watch it. It's very nice, and you will understand, or it's much better for visualization of those compartments, of those divisions. Okay, let's move further. Ribosomes. When we are talking about ribosomes, they are larger in eukaryotic cells than in prokaryotic cell. Here I prepared some picture for you. So ribosomes, we will get there as well today because it was a part of one pair for today. Here we are able to see prokaryotic ribosomes. You don't have to remember those subunits, numbers, and stuff. It's not important for you. What is important is that mm, prokaryotic cells, or almost everything in prokaryotic cells, is smaller or more simple comparing to eukaryotic cell. So here we have a ribosome of prokaryotes. Here is a ribosome of eukaryotes. You see functional 80S ribosome, functional 70S. So it's a little bit smaller. Regarding let me get back. So cytoskeletal components have not been found in prokaryotic cell. In eukaryotic cells, the cytoskeleton is always present. Yesterday we already mentioned that. Cytoskeleton is always present in eukaryotic cells. However, till this day, there was nothing like cytoskeletal structure found in prokaryotic cell. Okay? Proteosynthesis is much more complex, and also RNA synthesis is modified in the nucleus, and then proteins are modified in the cytoplasm. To all these things we will get. This is just, let's say, short recap of things and maybe a few more additional information to the table yesterday we were talking about. Okay, however, they are important. Yes, about mitosis. So The mechanisms of cell division is again different between eukaryotic cell and prokaryotic cell. Mitosis is the way or the process how the eukaryotic cells are divided. Okay. And yes, cellular compartments, we started already talking about that. So, because eukaryotic cells contain several membrane-separated compartments with different properties, the habit, let's say, of dividing the cell into so-called compartments has developed, or division is also possible, how to say it. In very broad sense, we can say that it's a cytoplasm with all the components and nucleus. We can say, if we are going into more narrower sense or more detail, each membrane organelle is a separate compartment. So as we were talking about Golgi apparatus, endoplasmic reticulum, mitochondria, each of those organelles, it's, let's say, different compartment or division. I do you follow so far, guys, about the compartments and the division? Do you understand what I'm talking about? Basically, what I want to say in this, let's say, more complicating way is that each organelle in the cell has its own function. And what then, if it's the own it has own function, so also it has also own membrane, so it works in different environment, different compartment on different division. For example, if you have a hospital, there is some surgery department, there is some neurology department, there is some dermatology department, hematology, and so on and so on and so on and so on. In all of those departments, they are doing something different, but they all belong to one hospital. Okay, the one hospital is cell, and it has different departments where different things are done. Okay, and we call it in the cell compartments. Okay, clear? Perfect. Okay, guys. Yes, we will discuss the endomembrane system, and then we will get to, no, we will get to the endomembrane system when discussing the cell organelles. Perfect, it's 8:48. Now I suggest whether you need a break or not. Okay, so there is a difference. So more people need a break than those who not. Perfect, guys. Okay, so it's 8:49. Let's meet at. Okay, sorry, it's my time. What is your time currently? Can you just very quickly tell me? Current time, Japan. You have 15:49, right? It's
Right.
yes, it's 15:49. Sorry. So at my time is 8:49. So let's meet at 15:58 here. Okay, let's have 10 minutes break. Hello, guys. Welcome back. So, after the break, I suggest you will have energy. So, let's start with your homework if it's possible. It would be great. Uh, Again, do not see the preparation time. It's not relevant for you. And however, we can start with the first pair, first group of people, first pair regarding endoplasmic reticulum. Here is just a optional picture. Nevertheless, do you have something prepared? Endoplasmic reticulum pair. Who was that? Endoplasmic. Yuiko, you are raising hand, right? Okay, and who was your colleague? Yuiko, who was your colleague?
Okay, what? Sorry, one more, please.
Yuiko, you prepared endoplasmic reticulum, right?
Yes.
And with whom? Who else? Because you were supposed to work in pair.
Oh, my peer is Mona.
Mona. Okay. Mona and Yuiko. Okay, ladies. Perfect. Have you prepared something in advance regarding endoplasmic reticulum? And do you have maybe do you have presentation or something? If yes, I can upload it, or you can share a screen if you want.
Oh no.
No?
Yeah.
Okay, so you will just read something, right?
Yes.
Okay, perfect. So we will use this picture. And yes, ladies, go on. What have you prepared?
Okay.
Just read what you have prepared, and we will then continue.
Okay. Okay. Endoplasmic reticulum have two types: rose endoplasmic reticulum and smooth endoplasmic reticulum. Yes. And rose endoplasmic is a system um, of membranes enclosing a fluid-filled space.
Mm-hmm,
And the surface is covered ribosomes
mm-hmm,
and food and processes proteins that have been made at ribosomes.
mm-hmm.
And,
Okay, is that it, or do you have something more? That's all.
No,
No? You are going, Mona.
yes,
Okay, go on, go
yeah.
on, go on.
Endoplasmic reticulum is a network of membranes inside a cell through which proteins and other molecules move. Proteins are assembled at organelles called ribosomes.
Mm-hmm.
That's it.
Okay, perfect. Okay, ladies, thank you very much. It was very nice. So let me provide you also and the others with some more additional information with endoplasmic reticulum. Thank you very much, Yuiko. Thank you very much, Mona. It was very good. Um, So, as you said, ladies, very good. We have basically two types of endoplasmic reticulum. Okay, we have a rough ER. We are able to see it here on this picture, rough ER, and and 1 second. I hear some echo. Now it's better. Yes, and smooth endoplasmic reticulum here. Okay, as you already mentioned, Yuiko and Mona, for those who haven't know anything about endoplasmic reticulum, we divide them. Misato, you can't hear me, Misato? I can't your voice. Guys, do you hear me? If you hear me, just raise your thumb up if you hear me. Okay, perfect. So most of you hear me. Refresh. Uh, Misato, refresh. Try to refresh. Sometimes it's problematic when you are joining from a phone. I guess more of you are using a phone. Guys, if there is something like this, try to maybe refresh. Many times it will, or in 90% of the times it will help that when you just refresh the website, it will work. So if you have some problems, try to refresh, it should work. More or less, this platform is very stable. Okay, let's get back to the endoplasmic reticulum. So, how to imagine, how to understand endoplasmic reticulum? Is an endoplasmic reticulum is an extensive network of membranes. Works now, Misato?
Yes.
Perfect. Okay. So I will just even now, yes, works. Perfect. Okay. So, endoplasmic reticulum is an extensive network of membranes that is directly connected to the outer nuclear membrane. Here on this picture, we are able to see a nucleus here, yes, and you see, maybe this picture will be better again. Here we are able to see nucleus, and it is directly connected to the endoplasmic reticulum. It surrounds the nucleus. Okay, let me get back. And so the ER forms a channel system of elongated cavities. Here it's nicely visible. Also on this picture it's nicely visible, those cavities, elongated cavities. And the most important function is synthesis of cellular components and cell export products. So, as you said, ladies, the ER is very important for synthesization of products which supposed to be within the cell or supposed to be outside of the cell. Okay, how the product will get outside of the cell? It's a part of exocytosis. Yesterday is very briefly mentioned the process when we are going from inside of a cell outside of a cell. Nevertheless, those particles here are important somewhere outside of a cell, and they are manufactured, produced in endoplasmic reticulum. So, guys, very simplified. If you have to write just one, remember just one thing regarding the endoplasmic reticulum, imagine it like a factory. Okay, endoplasmic reticulum is kind of factory which are producing some products which are necessary or very important for the cell itself or something outside the cell. Okay, so endoplasmic reticulum is a factory. Let's talk about more things which would be important for you. Rough endoplasmic reticulum. Why we call it rough, as ladies already explained, because there are more ribosomes, okay, on it. Rough endoplasmic reticulum, you are able to see it here, and smooth endoplasmic reticulum is here. Those small dots here around, and I'm pointing on them with my cursor, those are ribosomes, okay. So rough endoplasmic reticulum with ribosomes on the surface are very important for synthesization of proteins and then uh, uh, packaging those proteins and sending them to the Golgi apparatus. Okay, also proteins for cell organelles have their origin in rough endoplasmic reticulum. Uh, for example, as human cells example can be provided cells of pancreas. Are you familiar with the organ pancreas? Pancreas, have you heard about pancreas already? It's an organ in your body. Pancreas, there are plenty of enzymes, very important enzymes, for example, for digestion. And exocrine cells, those are just type of cells in pancreas. They are an example of cells which are very rich. They have like really big, those exocrine pancreas cells have very big endoplasmic reticulum, rough endoplasmic reticulum. Okay. Also, what is important to mention? Site of synthesis for peptide neurotransmitters and the presynaptic terminals. I said very, let's say, medical words. What does it mean? Neurotransmitter is nothing else. Like neurotransmitter is something to do with neurons, as you already maybe divide, and neurotransmitters are just substances or a way how the neurons are communicating. Okay, so let me just draw you something and I will explain it very briefly. So here we have let's say the end of one neuron. Okay, there is an body and it goes like here, and there is another neuron. Okay, so this is something what in your brain is going on. Okay, there is another neuron. A neurotransmitter is just this particle somewhere here which goes from this neuron to the second neuron, and this particle we call neurotransmitter. This is the way how the neurons are communicating. And those particles here are produced in in neurons and in their rough endoplasmic reticulum. Okay, so that's why we are talking about. That's why it is important, because there are plenty of diseases, plenty of cases which can affect this communication between neurons. Okay, for example, currently there are very increased tendency in developing mental health disorders or diseases. Maybe you already heard about depression. For example, depression is treated with antidepressants, and antidepressants affects and work directly on this communication here. Okay, they try to keep those vesicles for a longer time in the space between neurons, and that as an outcome will not decrease your mood, will not, you will not be so depressed. Okay, so this is maybe some one clinical correlation what we are talking about. That everything in medicine, everything in the human body is all, let's say, more or less connected. So that's why we need to know something about endoplasmic reticulums, for example. That was just one of million examples. Okay, let's talk something about smooth endoplasmic reticulum. As already ladies mentioned, so those are there are really less ribosomes on the surface, so that's why we call it smooth. And also we when we were one more additional information you will need for your histology part. Here in the rough endoplasmic reticulum we call something like Nissl substance is a stain or a color color, which you will pour on a sample of neurons, and the rough endoplasmic reticulum, after pouring this substance on it, will change a color. We use that in histology, and that's because of those ribosomes and the structure. So Nissl substance, you will need that not for admission, but definitely for histology. And I think yes, it will be. Not I think it will be in the first year. So something for your interests, if you are interested in. Smooth endoplasmic reticulum, on the other hand, is very important for biotransformation of drugs or alcohol or toxins. Can you guess, any of you, where would you expect to find cells which are very rich? They have a lot of smooth endoplasmic reticulum if we are talking about toxins, alcohol, drugs, and their transformation, their metabolism. Where would you expect them? Which organ in your body is very necessary for you to somehow metabolize toxins, drugs, alcohol, and so on? What are your guesses? Now?
Liver.
Perfect. Great. Very good. Very good. So hepatocytes, those are the liver cells. We call them hepatocytes, are very rich for smooth endoplasmic reticulum, exactly as you said, because so one of their feature is that their smooth endoplasmic reticulum is much more bigger comparing to the other human cells. That's because they need it. They need it for biotransformation and metabolism of those substances we mentioned. Okay, so very good. So that was it, guys. I would say everything what was necessary to mention about endoplasmic reticulum. Okay, so we were talking about this organelle. Did you copy the information? Was it okay, or was it overwhelming already for you? Was it okay? Doable? Can we continue? Perfect. Okay, if you agree, please just note, okay, or raise your thumb up, and you don't have to unmute yourself. Just some non-verbal communication, it would be great. Thank you very much. Okay, so it was Mona and Yuiko with endoplasmic reticulum. Who is with Golgi apparatus? Now and Misato. Perfect. So you can start.
Golgi apparatus consists on a flatted disc with overlapping single membrane patches and a surrounding spherical structure. And Golgi apparatus is responsible for transporting and modifying and packaging proteins and liquids into vesicles for delivery to targeted destination.
Mm-hmm. Very good. Thank you.
That's all.
Thank you. Now, Misato, do you have anything more? You are muted, Misato. Uh, Misato, you are muted. I have to unmute you. Oh, I can't.
Ah, okay.
Yes.
I think Golgi apparatus has a role which modify and secrete protein.
Mm-hmm.
That's all.
Okay. Exactly. So, guys, we were talking about endoplasmic reticulum as a factory. So let's imagine Golgi apparatus as a post office. Okay, post office. So it's packaging. Okay, it's packaging the vesicles and the products of endoplasmic reticulum mostly, and it delivers. Okay, so Golgi apparatus decides, not it decides, but it delivers whether it is going somewhere inside the cell or outside the cell. Okay, so post office. As you said, ladies, the Golgi apparatus functions as a molecular assembly line in which membrane proteins undergo extensive post-translational modification, basically. So it modifies after a translation. Translation is a process when the part of proteosynthesis. Translation is the last step of proteosynthesis. Basically, proteosynthesis means it's synthesizing proteins. So when your cell is creating, producing proteins, okay, its last step we'll get there. It's translation. I think in 3 days or 2 days we'll get there. Uh, so this is one process, and we are now talking about final product, some kind of protein which is necessary. Golgi apparatus, it modifies. It modifies. So it do some changes inside, okay, it modifies it, and Golgi apparatus package that and sending somewhere inside the cell, outside the cell, so post office. Golgi apparatus is post office. To remember that. Many Golgi reactions involve the addition of sugar residues and to membrane proteins and secreted proteins depends on the needs. So Golgi apparatus or those modifications can have basically some addition of sugar or carbohydrates to that. Okay, so yes, what else to mention? Carbohydrates. And yes, on this picture, we are able to see how we divide, or yes, how do we divide the Golgi apparatus. In basically, uh, this is all complex of Golgi apparatus, and there is a cis Golgi network and trans Golgi network. Cis Golgi network is closest to the ER, and the cis Golgi network is uh, accepting the delivery from ER. So basically, imagine it like ER. Oh, not sorry, I am confusing you with abbreviations. ER is endoplasmic reticulum. Endoplasmic reticulum is a factory producing some product. This product goes to Golgi apparatus firstly through cis Golgi network. Okay, so it's like a first step. Then here in the middle part, it's modified. Okay, modified. So it's adding some carbohydrates. Depends on what on the need. So there is some modification, and through the trans Golgi network, it's sending or delivering where it is needed. Or what was already said, outside or inside of the cell. Okay, so those are two parts, and yes, also it depends. For example, the stations where it can be delivered inside of the cell can be cell membrane, lysosomes, some storage vesicles, or other organelles. Yes, that would be it, I think, for your stage for now, how to understand Golgi apparatus. Okay, so thank you now. Thank you, Misado, for that. And let's move then further to lysosomes. Who covered lysosomes? Satsuki, you are raising hand. Sorry, I didn't see that. Satsuki, and who else? Satsuki, can you unmute yourself and tell us who was your colleague?
My partner is Aoi.
Aoi, perfect, Aoi. So Aoi and Satsuki, you prepared something for lysosomes. Perfect. Can you start maybe? Tell us something.
Okay. Lysosomes are single membrane vesicles containing about 70 hydrolyzing enzymes. Waste products produced in the cell are encased in vesicles, which fuse with the lysosome membrane. The hydrolytic
Hmm. If the cell is damaged beyond repair, lysosomes can help it to self-destruct in a process called programmed cell death or apoptosis. That's all.
Perfect. Do you know what apoptosis is? Or anyone? Apoptosis. Apoptosis, guys, is a programmed cell death. Okay, so when there is something wrong with the cell itself decide to kill herself. Okay, so it's some kind of suicide of a cell, and it's absolutely physiological process. It's absolutely normal. Apoptosis, on the other hand, is something the cancer cells doesn't have. Okay, so cancer cells, even though they are wrongly functioning, they are wrong, they are sick, let's say, they do not have this feature, so they are not killing themselves. So that's why are they reproducing and multiplying, and then why are they reproducing so quickly? Because they do not have this feature, uh, and uh, classical physiological cell, which everything is healthy, can kill herself when something is wrong. So it's absolutely normal. So that's the apoptosis, programmed cell death. That's everything you have to know so far. There are really complicating pathways how the cell trigger the apoptosis. For you so far, it's not relevant. But apoptosis as a term, you can already comprehend what does it mean, and it's physiological thing. Let's get back to lysosomes. Lysosomes. How do you think, or how would you imagine the lysosomes as an organelle? We heard from Satsuki and Aoi that are some hydrolytic enzymes. So hydrolytic enzymes are somehow handling with, for example, proteins or organelles or some objects, let's say, or particles in a cell which are not necessary anymore, and they try to metabolize it somehow, and also, um, sorry, that's my bad. Like dismantle or apart to pieces, okay, break them up. How would you imagine lysosomes in your own words, or to a 4-year-old child? Lysosomes. What comes to your mind? You are breaking the things up. You are breaking the things up which are not relevant or not useful for the cell anymore. Where would you put this waste? In a garbage, let's say, or rubbish dump. Okay, so um, imagine lysosome something like a trash dump, where or trash land or organelle, where everything what is not already or useful anymore for a cell, it goes to lysosomes, and lysosomes has to deal with that. Okay, so imagine it like this in a very simplified way. Do you understand the term trash or rubbish? Yes. Do you understand? So basically, you have some garbage. I don't know. You were cooking. You were cooking home. You used I don't know, meat, potatoes, and there are some packages from it. You don't need them anymore because you already cooked. So you put those packages to your garbage dump or trash dump. Basically, this is something similar as lysosomes are. Okay, but lysosomes can handle it very good. Okay, so they can metabolize it. So there is really just not a so big waste of things. They can nicely recycle the products. Okay, so as we already heard from Satsuki and Aoi, there are some hydrolases. Hydrolases are hydrolytic enzymes, as you already said, and then those hydrolases can digest proteins, nucleic acids, lipids, or complex carbohydrates. What is also very important to mention regarding lysosomes that its lumen is more acidic. Acidic. Are you familiar with the term acidic? What does it mean, acidic? Acid. Acid. Um, let me try to find a translation or maybe explain it in English. The pH of it is lower. However, I don't think you are familiar with the pH already. Um, it's more acidic. Um, is there anyone who is familiar with the acid or not? Acid. Yes? Sora, you are nodding. How you say acid in Japanese?
Acid, it means sanse, sun, I think, in Japan.
Okay, guys. So san, you said
Sanse,
sanse.
san.
Okay. So it's basically something very acidic or sanse, and you can metabolize, hydrolyze, break the things we mentioned up, so there will be not a lot of waste inside the cell. Okay, we call it in English acidic. If you have cytoplasm, there is some we describe the environment whether it is acidic or basic or alkalic. And the what is important is the pH. pH is basically how do we measure. Sorry, Gordon. Sorry, my dog decided to play just now. Uh, Okay, so and we have acidic or more basic or alkalic environment, and we measure it by pH. Okay, physiological pH for you so far is to remember pH is something around 7.4. Okay, so this is more or less the average pH, which is physiological. If we are talking about acidic, the pH is much lower, okay? So it goes down. And the lysosomes pH, or the concentration of the acids, is the pH is very low and the concentration of acids is much bigger. Okay, so there are plenty of acids which can break the already mentioned structures like nucleic acids, lipids, carbohydrates up. Okay, so it breaks them up. So this is the important. Lysosomes, in the other hand, or in the other words, can be called as acidic vacuoles if we are talking about plants or fungi. Okay, in plants or fungi, we have acidic vacuoles. On this picture, we are able to see a vacuole. Okay, this big thing is vacuole. So, but um, the function itself is very similar to lysosomes. Okay, okay. How do we get the things to lysosomes? Is basically from endocytosis. You already saw what endocytosis from outside the cell, somehow it gets to inside of the cell, and if it has to be metabolized and wasted, it goes to lysosomes, or it gets there directly from Golgi apparatus. Okay, from Golgi apparatus, some proteins are delivered to lysosomes. If some proteins were, for example, wrongly manufactured or produced, or for example, for some necessary structure of lysosomes, because also lysosomes has its own structure and has lysosomes have their needs. Their Golgi apparatus is sending and delivering those proteins to lysosomes. Mhm. Um, okay, okay, okay. I think this is most important part. Oh, sorry, this is lysosomes part. So as you see here, we have enzymes, membrane, and some transport proteins. Okay, okay. What about next part, which is which are ribosomes? Ribosomes was it? Yes, ribosomes. Who had ribosomes? Sora and Shoko. Perfect. Go on.
Yes, I will explain about structure, and Sora will explain how it works and effect.
Perfect.
So ribosomes doesn't have membrane structure and present in all cells. And ribosome synthesize proteins outside the nucleus.
Hmm.
And two large and small protein attach and make a shape of ribosomes, and each part is called a subunit,
Hmm.
and the size of the two subunits varies from organism to organism. It is a complex made up of protein and RNA, and ribosomes are free floating in the cytoplasm or attached to the endoplasmic reticulum. That's all. So Sora. Sora.
Sora, you
Uh,
muted. No, it's okay.
wait,
Sorry. Next, I introduce about ribosome main point. I think ribosomes has three main points. First, microscopical particle structures. Next, synthesis with protein. Then made by nucleus, which its shape is like ribbon, putting a lot of ribosome there. Then, how to work ribosomes in our body? Particularly, it is transfer is big point to work in our body. Transfer is to make protein by mRNA information.
Mhm.
That's all.
Perfect, perfect. Thank you very much, Sora. Thank you very much, Shoko. It was very nice. So, yes, maybe I will just add, I don't think I will add a lot. Just as you said, ribosomes are very small structures that we classify as an organelles. Even though they have no membrane, as you exactly as you said, we can say they belong to endomembrane system, to endoplasmic reticulum, lysosomes, and Golgi apparatus, because they are part of endoplasmic reticulum. But it's very questionable. It depends on authors or scientists you are reading articles from, or textbooks from. So yes, as you said, ladies, very good. So, as you said, Sora and Shoko, both of you mentioned that it's important for translation. Yes, translation is a part of proteosynthesis. We will get there. Here is a nice picture. Let me show you this picture. This is a picture where there is a ribosome small unit, and Shoko were talking about two parts, or sorry, I'm not sure which of you were talking about, but here we are able to see those two proteins parts, small and big one. Okay, Um and here we have an mRNA, and the other type of RNA, tRNA, gets to the ribosome, and together with ribosome, they produce amino acids and they produce the protein. So this is basically a simplified, very simplified version how we get or how we synthesize the proteins on the ribosomes. Okay, so here we have a ribosome, some strand of RNA, and ribosomes just go through it with another type of RNA, and amino acid or protein is produced. Okay, and this is happening outside the nucleus. This is happening in the cytoplasm. Very good, you said. You explained that, ladies. Um, yes. Chemically, ribosomes is composed of protein components and ribosomal RNA or rRNA. We are not able to see it on the picture, but this rRNA is part of their structure. Regarding the RNA, maybe it's complicating already for you, because here we are able to see mRNA, tRNA, I mentioned just rRNA. We will talk about DNA and RNA very soon. I think at the end of this week or during the next week, so it will be more familiar. So far for you to know is that what ladies, what Shoko and Sora mentioned for you uh, about ribosomes. So we have two small units, and they are very important for producing proteins. Okay, so this is what it's important to mention, and also the thing we already mentioned before. Prokaryotic ribosomes are smaller comparing to eukaryotic ribosomes. Very good. Very good. Thank you, Shoko. Thank you, Sora. And let's get to the cytoskeleton and cytoplasm. Who had this part, please? Yuki, you are raising hand, and who else? And Yuta, perfect. Okay, so you can start.
Yes, the cytoskeleton organizes other constituents of the cell, maintains the cell's shape, and is responsible for the locomotion of the cell itself and the movement of the various organelles within it. Three major types of pyramids make up the cytoskeleton: actin filaments, microtubules, and intermediate filaments. Yeah.
Okay. The cytoplasm is the area of the protoplasm, the part of the cell surrounded by the cell membrane other than the cell nucleus. It is where most of the various metabolic and cellular act activities in the organism, such as cell division, occur.
Mhm.
That's all.
Perfect. Thank you very much, Yuta. Thank you very much, Yuki. Uh, Very good. You mentioned everything important. So let me just add few things regarding cytoplasm. Cytoplasm we are able to see on this picture here. So cytoplasm is or cytosol depends. uh, some some resources are dividing the cytoplasm and cytosol. I'm actually not a big fan of it, because I don't see the difference between those two terms is very significant. Nevertheless, it depends on the source. I will also, I would like to play maybe a video, or I will send you definitely a video um, where is comparing a plant cell and animal cell, and they, for example, are differing cytosol and cytoplasm, but more or less we are talking about the same thing. So cytoplasm is a fluid filling space inside of the cell membrane or plasma membrane, as Yuki and Yuta already mentioned. Um, okay. Why we are hearing the echo? Mm, okay, now works. Perfect. Uh, and it is permeated with cytoskeletal filaments. Very nicely, Yuki explained. We have microfilaments, microtubules, and intermediate filaments. Okay, guys, please zoom in a little bit to the picture. Here we have filaments, actin filaments, intermediate filaments, or microtubules. Please note that down if you are doing notes. Those three are very essential, and I guess already in admission questions they can occur. What kind of filaments or cytoskeleton structures do you know? Okay, very important. So, and cytoplasm, besides cytoskeletal filaments, there are also the organelles, ribosomes, cytoplasmic proteins, enzymes, soluble components. Among those soluble components, we count ions, glucose, waste substances, metabolic intermediates. So everything basically what is inside of the cell membrane, inside of the cell, is somewhere located in the cytoplasm. Uh, yes, and some. When we are going to talk about cell membrane, we will talk about membrane channels and also Na plus, K plus, or sodium potassium pump for maintaining an content inside and outside of a cell membrane. So this is also a part of a cytoplasm. And regarding the cytoskeleton, here we have nice pictures of it. So as one said, actin filaments, intermediate filaments, and microtubules. Actin filaments can be also called as microfilaments. This is another term very usually used, and they are different regarding the diameter, how thick they are. So as you see, actin filaments here are the narrowest, only 7 nanometers, intermediate 10 nanometers, and microtubules are 25 nanometers. Those thickness, you don't have to remember those numbers. Okay, this is just an example what is different among those filaments, okay, and tubules. And yes, basically, as Yuki and Yuta explained, cytoskeleton is something like it's structuring, it's giving a shape it's giving a shape of a cell. So we can call it as scaffolding, or do you scaffolding, are you familiar with with this word? Maybe when you have a building, when you have a building and you are starting from a base and then you are giving a shape. Scaffolding is something you are using for building a building. Okay, so this is a scaffolding, and the same, let's say the same function is for cytoskeleton. Okay, guys, so thank you very much also Yuki and Yuta. It was very good. Thank you very much that you all of you that you prepared something. And We will have to end soon. So that was basically it. Guys, I will upload one video for you. You don't have to prepare anything. Just for a summarization of animal versus plant cells, because I think tomorrow we won't have enough time to watch this video. Nevertheless, we covered everything from eukaryotic cell, and there are some differences regarding the plant cell. You can watch this video, I will upload it, it's very short, and that's everything you will have to know for tomorrow. And it would be great, it would be very good if you would watch the videos about inside the cell membrane and cell transport. Inside the cell membrane is 9 minutes long, cell transport is 7 minutes long. So together it's 16 minutes. It would be good if you would watch it for tomorrow's session. You don't have to study a lot, just to have an idea what we are going to do. So it's just 15 minutes. And as a recap for of today, the video, maybe some of you already saw that, the video of biology cell structure, which will be together 23 minutes. So if you will invest 23 minutes of your life or of your time to watching those videos, it would be very beneficial, very helpful for you. Okay, so here we will end. Uh, and yes, that's everything from my side. Thank you very much for active participation, and that you were very good prepared, and you have done pretty nice job today. So looking forward to see you tomorrow. Do you have any questions? No questions? Okay, guys, so I will upload one more video. If you will have time for that, you can watch it. Just for your interest, tomorrow we are going to do something about cell membrane and cell membrane transport. Okay, so those are topics for tomorrow. Thank you very much, guys. See you tomorrow. Have a nice day.
Thank you very much.
Thank you very much as well.
Thank you.
See you tomorrow.
See you tomorrow.